concept of the immortal DNA strand was put forth by John Cairns in 1975, suggesting a method through which adult stem cells could limit genomic mutations. Cairns posited that instead of dividing their genetic material indiscriminately during mitotic cell division, adult stem cells distribute their DNA asymmetrically, holding on to a distinct set of original, parental, DNA strands through each cycle. By maintaining these original strands, any mutations occurring from DNA replication errors could be passed on to non-stem cells, which then ultimately complete their life cycles by differentiating into functional cells. By transferring these errors, stem cells could curtail their own mutation accumulation rate, potentially reducing the risk of serious genetic diseases such as cancer. While supporting evidence for this procedure does exist, whether it plays out in vivo within adult stem cells remains a contentious issue. Two primary tests are used to track this hypothesized immortal DNA strand: label, retention, and label, release pulses chase processes. The label retention test aims to tag original or parental DNA stands with a DNA marker like tritiated thymidine or bromidioxyuridine, BRADU. These labels merge with the new DNA in dividing cells during the ES phase, a stage in cell division. Here, stem cells have yet to establish an immortal DNA strand, either dividing symmetrically, which means a new immortal strand is determined each cycle and will be labeled in at least one stem cell, or their precursors divide symmetrically, and once they mature into adult stem cells, the immortal strand that's already marked is chosen. In the experiments, stem cells divide symmetrically during growth and post-wound recovery, and it's uncertain during neonatal stages. Once their immortal strand is labeled and asymmetric division begins or continues, the DNA marker is traced. With symmetric division, most mitotic cells split their DNA randomly, and the DNA marker will become too diluted to detect after about 5 divisions. But if cells utilize the immortal strand mechanism, all marked DNA will stay with the adult stem cell even over 5 or more divisions, making it detectable within the adult stem cell. These cells are also known as label retaining cells, ARTCs. On the other hand, the aim of the label release test is to mark new DNA typically transferred to the daughter non-stem cell. This is done when stem cells are in asymmetric division. Given homeostatic conditions, adult stem cells should divide asymmetrically to keep the tissue compartment's stem cell count stable. After an adequate pulsing period to label all newly replicated DNA, this label is chased. Each DNA replication now incorporates unlabeled nucleotides, and the adult stem cells are tested for loss of the DNA label after 2 cell divisions. If cells are randomly segregating, enough DNA markers should stay in the cell to be detected. Scientists working with DNA have discovered a mechanism that enables adult stem cells to divide unevenly, known as the immortal DNA strand hypothesis. Initial experiments involved using multiple DNA labels, one to denote the immortal strands in stem cells, another for new DNA synthesis. The cells can retain one label and release the other after 2 divisions, implying an immortal DNA strand mechanism. Early pieces of evidence for this theory have been found in various systems, with one of the first studies initiated by Carl Lark and his team, where they noticed the DNA in plant root cells co-segregated. Despite not all DNA segregating, the amount seen in one daughter cell corresponded to the quantity resulting from sister chromatid exchange. Subsequent research by Christopher Potton and colleagues, 2002, used similar methodologies to discover long-term label-retaining cells in the small intestines of newborn mice. They believed the long-term retention of these labels was caused by the mice's underdeveloped intestines and neonatal period when immortal DNA was being formed. Importantly, these label-retaining cells were active, indicating they were using immortal DNA for division. Research from James Shirley's lab, led by Joshua Maroc, developed mammalian cells with an inducible gene controlling uneven cell division. Studies indicated that chromosome segregation occurred in a non-random manner only in cells stimulated to divide similarly to adult stem cells, offering an in vitro model for demonstrating the immortal strand mechanisms. This concept has been explored in other stem cell populations whereby, for instance, Gilbert Smith and Nick Zeeps observed label retaining cells in mouse mammary glands. At the same time, Derek van der Kui's lab discovered neural stem cells in mice that retain labels and are actively dividing. In 2006, the Shaharajim Tajbakush lab added to the body of evidence, showing that muscle satellite cells, possible adult skeletal muscle stem cells, exhibited asymmetric segregation when cultured thus. These studies provide intriguing evidence supporting the immortal DNA strand hypothesis across a variety of systems. Using the model of muscle stem and satellite cells during tissue regeneration, a period marked by rapid and substantial cell division, scientists have been able to observe an intriguing separation of old immortal DNA and new DNA among dividing cells. Approximately 50 of the dividing cells in regenerating muscle sort the older DNA to one daughter cell and the younger daughter cell and the younger DNA to the other consistently. The more undifferentiated daughter tends to inherit the older DNA chromatids, while the more differentiated daughter inherits the younger ones. However, experimental proof against the immortal strand hypothesis remains elusive. One study failed to find a consistent pattern supporting the hypothesis while tracking the release of tritiated thymidine in dividing murine epidermal basal cells. Despite identifying label-retaining cells, these were not situated within the purported stem cell compartment. In a study on DNA template strand partition in developing zebrafish, rapid depletion of older DNA template strands from stem cell niches in the retina, brain, and intestine was observed during larval development. No signs of asymmetrical template strand segregation were found, suggesting asymmetrical DNA separation does not prevent mutational burden in developing zebrafish as the immortal strand hypothesis proposed. The theory of immortal DNA strand mechanism, first suggested by Cairns, has since been refined. In 2002, it was proposed that mature stem cells will choose to perish, apoptose, when their immortal DNA strands suffer damage, instead of employing typical non-stem cell DNA repair mechanisms. Tannenbaum and Shirley developed a quantitative model highlighting the enhanced efficiency of DNA repair in adult stem cells utilizing an immortal DNA strand mechanism for DNA segregation, as it prevents the tendency to mistakenly fix DNA mutations in both strands, thereby propagating the mutation. Although some critics have challenged this work, the authors have defended their findings robustly. The theory is supported by the suggested mechanism of the dynein motor, adding credibility to the concept despite its contentious nature. While teased apart by diverse studies and arguments, the immortal DNA strand hypothesis remains a fundamentally intriguing concept in the realm of biological science.